Hi, we are Group DUS, Marisa and Abigail, and today we're going to be talking about our gene, which is Bradi1g65010. So we'll be talking about how we explore this gene structure and function, um, and it's a gene in Brachypodium Distachyon.
So for our gene, we made two gene models. The first was the ab initio model, and this is a predictive model. It showed that we had 8 exons, which is represented here, and then in between we have 7 introns. And then for our homologous model, which is based on structure, we also had 8 exons and 7 introns, but exon 5 was significantly longer, and this resulted in a longer coding sequence. And overall, our homologous model was a better representation because it had a longer coding sequence, it was based on structure, and it had more coding potential.
So using our models, we were able to perform a literature research to find out more about the function of our gene. So the BLAST results showed that our gene produces a protein, which is a member of the SCAR or WAVE family. And it also said that this family of proteins has a lot of significance in actin cytoskeleton organization. So performing a literature search on our gene and that particular protein family, we found that the SCAR protein family is important in the activation of complexes which are important for actin nucleation. So this is really important for cell structure and function because it's how actin polymerizes and forms these exoskeleton structures for the cells. We also found experimental results which showed that mutating the genes coding for SCAR and WAVE protein impaired the ability of these plants to grow. So they had significantly less growth and strength, which is measured by agar penetration.
So in our gene, we identified the NAN1582 mutation, which is a high impact nonsense mutation located in exon 5. Here you can see the location and position. And so what this mutation does is that it changes the guanine base to a thymine base, and this causes the amino acid to change from glutamine to a stop codon. This is known as a stop gain mutation, and it's high impact because the early stop codon would cause a significant loss of coding potential.
So we performed a sequence of protocols for DNA extraction, quantification, and PCR. And we designed primers to isolate that mutation within exon 5 so that we could amplify this region and sequence it. So our final product, based on the primers we designed, should be around 750 base pairs. And we have the gel on the left that shows the sequences run in an agarose gel electrophoresis once they've been sent through PCR with these primers designed. So you can see that they're at about the correct location based on our ladder, about 750 base pairs, which indicates that we successfully isolated these fragments through the PCR procedure. We were able to extract these bands from the PCR and send them for Sanger sequencing.
So for our Sanger sequencing results, out of the 3 samples we sent, only one of them was a homozygous mutant, and the rest were homozygous wild types. And so for the first chromatogram, you can see that there's no noise indicated by the clear peaks, and the mutation, which changes the guanine to a thymine, is highlighted in the pink box. And this causes a gap in alignment in the sequence, which is shown in the orange box. And so basically, this proves that the mutation exists.
So we were able to perform a phenotype analysis based on the Sanger sequencing. So we were able to compare cross sections of the first internode of sequences from plants that we knew were mutants versus wild types. And we were able to identify them as mutant or wild type from those Sanger sequencing results. And because we wanted to identify them as mutant or wild type, we were unable to get a lot of pictures because we only had one plant of the mutant form. So you can see where we were able to get pictures of the mutant and wild type, but they were of very limited quality. because of the quality of the cross sections and the limited number of plants. So we weren't able to draw definitive conclusions from this phenotype analysis.
So, in order to first design an experiment, we had to figure out where our gene was most highly expressed. We did this by looking at an electronic fluorescent pictograph of the Distachyon, and this revealed that our gene is most highly expressed in the stem and roots. You can see this in the figure, which shows gene expression from a scale from yellow to red, red being the highest expression. And specifically, we see that the highest expression is in the first internode, indicated by the red color, and then the second highest expression is in the roots, indicated by the orange color. And this supports our hypothesis because it shows that it's involved in growth and structure.
So we were able to perform data analysis based on data given to us from other experiments. And so we were able to perform this data mining in order to draw further definitive conclusions about where our gene is expressed and then potentially about gene function. So we were able to see highest expression significantly in the roots and stems versus the leaves. And then in the other graph, we see lower expression in the leaves compared to the flower. And so typically a lot of this growth is happening in that first internode, in the stems and in the roots, which supports the conclusion from the literature search that it's involved in the growth and the structure of the actin cytoskeleton.
So our experimental design, we used gibberellic acid, which is a growth hormone, and then we had a control group, which was just water, and we used this to test the internode growth of our gene expression. And specifically, we had the 3 groups in soil and then the 3 other groups in soil for the control and experiment. And the experiment was treated with 10 micromolars of gibberellic acid. They were left for 2 hours, and then we collected a sample from the internode, and then we compared the gene expression in the 2 samples. We believed, if our hypothesis was correct, that we would see more expression in the internode that was treated.
Right, so we collected samples from both treated and untreated samples, but due to predicted problems in the extraction procedures, we got low concentrations of RNA for all of our samples. And we see high levels of contamination as well, as indicated by the absorption values. In the gel we ran of these samples, we only had 2 lanes that had the distinguishable RNA bands. So this is likely the result of poor extraction procedure or errors while loading gels.
So next we did an analysis of the qPCR results in order to identify our gene expression. And unfortunately, we didn't really get much results. And that's likely because our RNA extraction didn't go so well. But you can see that control and treatment is our samples, while the wild type and the mutant is the provided samples we received from Dr. Hazen. And we weren't really able to make any conclusive comparison between the gene of interest and the housekeeping genes just because there wasn't enough data. But we did make one graph showing that there is a higher relative expression in the control group compared to the treated group. But there's nothing really conclusive about this because we basically only used like one data set, and this is not representative of the data.
So all in all, we weren't able to make definitive conclusions from most of our analysis, but we did have some successful procedures, and we were able to support our hypothesis about gene function through some of the data mining that we did. And so this data mining showed high expression in roots and stems, which corresponds to our predicted gene function that we got from the literature search. So this predicted function, again, was that our gene is important in actin nucleation and activating that complex, which allows for polymeration of the cytoskeleton and the actin filaments and able to help with cell growth and cell structure. So if we were to do more replicates of these experiments, we would expect to see more definitive results. But thank you for a great semester.